Good afternoon. I'm Ellie Walmark, and welcome to the ICM Research in Focus podcast series. Today, I have with me Alecia-Jane Twigger, Pedro, Victori, Rosa, Akanksha Anand, and Kui Hua, and Joe Worley, who are going to talk about a project that was funded by Wellcome LEAP under the Delta Tissue Programme. I'll ask them to give a bit more detail, but fundamentally, the aim of the project was to build a tissue time machine where they would characterise tumour samples from triple-negative breast cancer patients before and after treatment to build a model to be able to predict how they would respond to treatment. Hi, everybody. Thank you so much for joining me this afternoon. First of all, I'm just going to go around everybody, and if you could just tell me who you are and your job title and your affiliations, please. So, Akanksha, let's start with you.
Hello, so my name is Akanksha Anand, and I'm a postdoc in Greg Hannon's lab at Cancer Research UKCI.
Thanks. And Kui?
Hello, my name is Kui Hua. I'm a senior research associate at the CRU UK Cambridge Institute. I'm a bioinformatician.
Thank you. And Pedro?
Hi, I'm Pedro Victori. I'm a postdoctoral research fellow in computational cancer at Jasmine Fisher's lab in the UCL Cancer Institute in London.
Thank you. And Alecia-Jane?
I'm Alecia-Jane Twigger. On this project, I was a postdoc working between the Stem Cell Institute and pharmacology. Now I'm a PI in pharmacology and biochemistry.
Congratulations.
Thanks.
And Joe?
Hi, I'm Jo Worley. I'm the Breast Cancer Programme Manager for the CRUK Cambridge Centre, and I work with the Precision Breast Cancer Institute team, and which is led by Jean Abraham.
Brilliant. Thank you very much. So first of all, I'm going to ask each of you to briefly outline your role in the project. So let's go around the same way. So Akanksha, if you start.
So my role in this project was more catered to sample processing, tissue collection, and then using those samples for various technologies such as single nuclear RNA sequencing, single cell RNA sequencing, image mass cytometry, and some spatial genomics. This included data generation as well. And apart from that, I was also involved in the pathology analysis of these human tissue samples.
Brilliant. And just tell me and my listeners what mass spec cytometry is.
So image mass cytometry is basically using mass cytometry and image analysis and combining these two. So what you get is you take a tissue sample, you stain it as you do with a regular immunohistochemistry, but the difference is that you use metal-based antibodies, so that when you run this through a machine, which basically does mass spec, those metals are charged and ignited, and those are laser captured. So that actually tells you the position of the protein on the tissue itself. So you can visualize it, and you don't just get graphs as you get in mass spec. So in total, that's what image mass cytometry is.
Thank you. That's a brilliant explanation. And let's come to Kui.
My role is basically the bioinformatician of the team. I do all the computational analysis and integration across all different modalities. My role involved designing and implementing all the bioinformatics workflows for different modalities Akanksha mentioned, including extracting signals from those data and providing some basic interpretations, which can be discussed across all the teams.
Thank you. And Pedro?
So I was in charge of building a computational model of TNBC. This is based on previous work by the Fisher lab, and I worked in close collaboration with the rest of the team to ensure that my model included all the relevant pathways in the response to treatment in triple-negative breast cancer. And then I created what we call digital twins by initializing my model with each specific patient genetics and ensure that my model for each of these patients could reproduce the response that the patients showed. And after this, I focused on using the model to predict new strategies for treatment that could resensitize patients who are not responding to standard of care therapy.
Wow, that's amazing. So you made a digital copy of each patient with your model, essentially.
Yeah, so to speak, yeah.
Wow, cool. And Alecia-Jane?
Yes, so my job was kind of sidekick to Akanksha, where we were processing the samples actively together. We had to optimize all the protocols and figure out how we could take a tiny piece of tissue and do all of this omics analysis on it. I also had to wrangle PIs as well as core facilities and basically work across with everyone to make sure that the project was on track, making sure we're hitting the key milestones of the funders and delivering what we promised.
So you were the magic that made it all happen, the glue that stuck it together.
I'd say we were all the magic that made it happen, absolutely.
And Jo?
So my role was a little different. So I think what made the project quite unique was that we were using tissue samples that were collected from the partner clinical trial and also the personalised breast cancer programme. So my role was really kind of facilitating that access. But also we had to sort of work with the R&D department at Cambridge University Hospital and the university to make sure we had all the right consents and ethics in place to do that work as well, really before any of that could start. There was also working with our patient-facing team. So some of the samples were retrospective that had already been collected, but we also made a decision to collect some fresh samples. We worked with our patient-facing team in the Precision Breast Cancer Institute for that, as well as Tissue Bank. So yeah, I was kind of the link with everything that happened over at the kind of Cambridge University Hospital for making the project happen.
Brilliant. And that actually brings me really well onto my first question, which is where did you get the samples for this project? So did they all come from the Personalised Breast Cancer Programme and partner clinical trials, or was it further afield than that?
They were all partner and mainly partner and Personalised Breast Cancer Programme.
And are those clinical trials based in Cambridge, or are there different geographical locations for them?
So partner is a multi-site clinical trial, but because they're led in Cambridge by Professor Jean Abraham, and we have a lot of support through the Precision Breast Cancer Institute to do a lot of sample collecting. Pretty much all of the samples were from patients that were recruited in Cambridge.
And so what analysis did you perform on the samples and how did you integrate the data? Obviously, I'm from the Integrated Cancer Medicine Programme, so I'm really interested in how you integrated the data and why you thought that was important. Who wants to take that one first? Alecia?
I'll start with what we did, and then maybe Akanksha can jump in, and then Kui can say how it was analysed. What we did is we took these very small tissue samples that were either snap frozen or freshly collected, as well as FFP tissue sections. And from the small frozen samples, we then extracted nuclei, where we then conducted single nuclear RNA sequencing as well as single nuclear DNA sequencing. In complement to that, we would have spatial transcriptomics done on the FFPE tissue sections, as well as the image mass cytometry that Akanksha's mentioned. Now, from the fresh samples, we handled them slightly differently so that we could get out robust single cell data. So we did a partial dissociation on them, and then we processed them for single cell RNA sequencing, but with the added benefit of being able to do BCR and TCR sequencing on them as well. As well as sending them to Canada for single cell DNA sequencing as well.
Wow. So hang on 1 second. So you've used quite a lot of terms there. So tell me, what is the difference between all of those different types of sequencing?
So basically, we're trying to look at the genome, so the DNA level, the RNA level, and the protein level, and looking at on a single cell level as well as spatially. So how is the heterogeneity of the tissue looking? And then how is that kind of spatially distributed across a tumour sample?
Thank you. Great explanation. Sorry, and carry on.
I'll pass over to Kui in terms of the integration.
Thank you. So in terms of, I will start from the data analysis. There are different levels of analysis. I would say the first level is a quality control of the data. So as Alecia-Jane mentioned, we're using cutting edge technologies on those real world clinical samples, which is very challenging because we're doing a lot of things with very limited materials. So the quality is, I mean, the data is amazing, it's great, but the quality is just not perfect. So you need to guarantee that you achieve high technical quality data. That's the first step. Those including, removing noise, how to pre-process all of them. And next is the foundation of all the downstream analysis. That would be the basic annotation, biological annotation. For single cell data, that would be for each of the cell, what cell type are they? What cell states are they? And for spatial resolved data, that including each of the cells, where are they located? What areas are they located? What's surrounding them? This is the foundation. And the next level, it's a comparison. You want to compare those things between different conditions. For example, between responders and non-responders, do you see one of the cell type more abundant? Do you see they co-localize with a different cell type in the same way or in different ways? If a certain cell type or a group of cell types, they co-localize with each other, what are they doing? What genes are they using? Do such signals differ between groups? That would be the third thing. And the last step is to provide some sort of interpretation. That's the most important step. We have a biological interpretation of what goes wrong inside of the patient and what can be used as a marker or as a target eventually to treat those disease. That would be the analysis at different levels. So in terms of integration, that's even more complicated. So I would say at a higher level, integration, so first of all, each modality, they provide data from different angles, but no single modality is perfect. They give you partial data, imperfect data. So the first integration is to confirm some of the observation, are they consistent across different observations? If you see more abundant cell type A in responders, do you see them across in single cell data, in spatial transcriptomic data as well? So this is consistency you need to check. And more importantly, because each modality is imperfect, so we want to combine them all together to find something that each modality on its own would be impossible to tell you. For example, some H&E images, combining H&E images, spatial transcriptomic data, and the single cell data that would tell you where exactly the boundary between tumor cells and stroma cells is and what is happening there and how these things differs between groups. And this is really the signals that enables to give us some interpretations and predictive models for the downstream things.
When you say responders, do you mean which cells are responding to the treatment or which patients are responding to the treatment and which aren't? And you can tell that by looking at this single cell data.
I'm talking mainly at the patient level. So some patients we know from clinical definitions whether are they responding to the treatment. And then we zoom in to the cellular level and tissue level to see where it goes wrong. So, but you raise a very interesting question. At the cellular level, can we tell whether that's responding or not? I mean, the answer to that is it's complicated. I would say potentially yes. So some non-responders of the patient, the very reason is some of the cancer cells that responded to the treatment, but there are remaining cancer cells still there, making this patient a partial responder. So that's a very professional question.
Thank you. I try my best.
I would like to. I would like to add a couple of things here. So when we say we collected samples, we need to be mindful that we collected paired samples. So for a particular patient, we collected pre-treatment as well as post-treatment. And that is what helps us to put each patient differently, analyze each patient differently, as well as group them together. Then these groups were further divided into pathological complete responders and pathological non-responders. That's what Kui was talking about the response. And another thing that we understood during our project here was when we are dealing with human tissue, we face the challenge, and this is a challenge that will be faced by any human study, will be of limited tissue samples. The limited tissue that we have, because the priority will be the diagnosis of the patient. So that's why it was, and as Alicia mentioned, and she emphasized very much tiny piece of tissue. The reason for tiny piece of tissue and bit scattered data was like we are limited by the amount of tissue we get for our actual research purposes or for actual analysis. So this is something that is a holdback for any kind of human study, not just this one.
Yeah, that makes complete sense. And then once you've got all this information, I guess then, Pedro, it's over to you to put it into your model and see what that can tell us. Is that right?
Yeah, no, the multimodal aspect of this project was very valuable for me because I could integrate data from different layers of information into my model, which means that my model is not limited to only predicting things caused by mutations, for example, but I can also take into account, for example, large genomic events like copy number alterations and post-translational modifications, all kinds of molecular processes I can take into account in the model thanks to this multimodal aspect. And thank you to the work of Kui and the rest of the team who have analyzed it and given me the main insights.
And then you use all of this data to train your AI model, is that correct?
Well, I mean, technically, the model is not an AI model, so it's rather what we call a mechanistic network model. So we build them by hand by drawing all the interactions between genes or proteins. But basically, yes, I have taken all of this information to do the training of the model, but the training is me who has done it, and instead of just inputting it into an AI, there's a technical difference.
No, that makes sense.
Yeah.
And then how does the model that you developed then work to predict patient outcomes? Can it predict patient outcomes, or would that be a future use? Yeah.
It can. I mean, it can with, it has a certain margin of error, but basically, as I was saying, the model is a mechanistic model, which means that for every prediction of the model, we can look into why the model is saying that. So, like machine learning or AI sometimes can be a bit of a black box in that we know the answer that it's giving us, but we don't know how it has reached that answer. But because in my model is a transparent mechanism where I can see, okay, this gene is being activated in the model because this gene is activating it and this gene before it. So I can look at the whole sequence of causality. And so the model, we initialized it with genomic data from each of the patients. And in doing so, we are creating what we refer to as digital twins. Then for each of the digital twins, we apply the simulated treatment that the actual patient got. So patients who got chemotherapy or patients who got chemotherapy plus the targeted drug olaparib. And then I look at the output of the model, which in this case is predicted cell death or predicted cell proliferation. And I see if that matches the clinical real response of the patient. So for responder patients, I expect the model to predict a decrease in cell proliferation because the cells are dying and reproducing less, and an increase in predicted cell death.
That's really, really interesting. And so how long was the project that you took part in?
I think in total was 3 years. I joined in the second year, so for me it was 2 years, although I feel it was much longer because we did so many things.
Sure. And then, so what stage of the project are you currently at? Is it being used in a clinical setting now, or tell me about where you are now? Alicia, do you want to take that one?
Sure. So I have to say, the project finished in September last year, and we're still wading through a lot of the data. In terms of it going into clinical use, I think this is something that would be very far down the line, just because obviously anything that goes into clinics needs to be thoroughly vetted. The amount of data that we've generated as part of this project, it's not realistic to be able to collect that amount of data per patient in a clinical setting. So what we're hoping to do is, from the data that we've analysed, is to extract out key features that can then be used with more simple diagnostic tools that we could then take forward.
And what impact do you think that this project or the research coming out of this project could potentially have on patient treatment outcomes? Who wants to take that one? Akanksha?
So we would like it to have a huge impact, a huge influence. At this stage, the direction in which the medicine is going towards personalized treatments I think this kind of study and this kind of data will be very helpful in pushing it more towards patient care and personalized medicine. And this is something we've also seen in our data set as well, that each patient is different. And you cannot group all the patient in one huge sphere. So how each patient is different and why a certain treatment will work on a patient and not work on some patient, I think this study is kind of the groundwork of where we start understanding like where exactly two same patients with same disease start to become different.
Yeah, that's really interesting. Yeah, I can see that it could be used to understand why the treatment works for some and why not, and if there's any underlying things that you perhaps wouldn't know about the patient without doing all of these multimodal data analysis. Yeah. And then do you think, is it possible that it will become standard of care, this way of treating patients and looking at their outcomes and whether they respond or not? And my other question, part B to that, is can you do it outside of Cambridge? Can you use it in other geographical locations?
I think so, yes. I think this question has come up in a lot of our meetings before as well. And I think the idea would be to start using it first in-house and then start to propagate it, because that's the whole point of having a model and understanding the disease in a deeper manner, so that you can actually have multiple applications rather than doing same studies in different demographics and different populations, and like take this as the basis and then continue the application bit of it.
No, I was just going to add that for me, ideally, this method will allow to better stratify patients. So maybe we can't sequence each one of the patients, because that will be like very expensive. But we will be able to stratify them into groups where we can give each of them the treatment that is most likely to benefit them. So we can reduce, like very long treatment schedules and instead be more targeted and more precise. So we can also improve like patient quality of life by giving them things that actually are going to work quickly and in a precise manner.
Yeah, and by stopping things that aren't going to work, so you stop that extra toxicity in their body, I guess. Yeah,
exactly.
Yeah.
I think the other thing is, what hasn't really been explored yet would be how you could use some of the data that's been collected, or some of the targets that are identified in this study could then potentially be integrated with what we are more familiar with the kind of integrated cancer medicine. So the radiomics and the H&E, digital pathology images, that kind of information as well. So there's potential to kind of integrate that kind of cellular tissue level data with the sort of more macro data that we're also working on in the PBCI as well.
Yeah, absolutely. And do you think that this model could be used with different types of cancer, with other types of cancer, or is this very specific to breast cancer?
So no, actually we can use it for different cancer types. So in my lab, in the Jasmine Fisher lab, we have developed what we refer to as a library of models. So we have a model for a lot of different cancer types. So we have, for example, for glioblastoma, for lung cancer, for melanoma, and also this approach has been used for other diseases, other than cancer. So we use it for COVID, for example, a few years back. So yeah, it's a very flexible approach.
Yeah, that's great. So I know a lot of patients have concerns about their data being used by AI or models or, things that the researchers invent. How have you overcome the challenges of using real world data and keeping it all safe?
Because the model is running in our secure computers, no patient data leaves this computer. So all the data is secure and we are not using it for anything else. That is not what the patient has consented to.
Alecia-Jane, did you want to say something else?
Yeah, I was just going to say, I think Joe and Jean's team has just been fantastic in making sure that Every sample is anonymized, so we have no idea what patient sample we're working with. We've only been working on data sets extracted from genomic data, so we don't have any way to basically assign the data back to the patient at all. We take the ethics very seriously. We respect the patients, and we want to make sure that they have confidence in what we're doing. Yeah, we have no idea who they are, but we do thank them a lot for participating in our research.
I'd also add that we made sure that there were data and sample transfer agreements, even though a lot of it's within Cambridge, even within across the different departments, and also with Jasmine Fisher's team and the other teams involved in the project as well. So there was, that sort of official understanding of transferring data. And I would add that I think the way Alecia-Jane and Akanksha really managed and handled the samples, they kept really amazing records of exactly how those tiny samples were being divided up and used. And so I think that level of record keeping was really amazing.
Kui, did you want to say something?
Yes, I think some of the concerns, all of what the team has said, that's the key to guaranteeing they don't need to worry. Another thing that I think might be relevant is I think many of the concerns come from first, now a lot of AI tools have become so powerful, so people are just sort of afraid of that without really understanding what AI means. The reason why current AI model like ChatGPT is very powerful, it is because it basically has seen all the things on the internet, so sort of memorize all of that. And that's why it seems to be very powerful. Till now, the AI thing is still extremely powerful too. It's not magic. That's one thing. And secondly is for medical studies, especially those molecular and cellular studies, it's way behind the AI studies in those imaging and large language model is way behind that. And it's a long journey. It's sort of related back to your question when you're asking, do we expect it to have some like a revolutionary impact? We want to say yes to that. But meanwhile, we also understand that it is a long journey. We believe we will make some impact, but it's not like a single study would just resolve all these questions all at one time. And putting all this together, so it's not like we're sequencing some single cell data and feed that into AI model, and then magically it can knows everything about you. It's just not getting there yet. Not only we're not getting there yet, we're still struggling to understand the difference between responders and non-responders. At a population level, we're still struggling, let alone at the individual level. So I would say the journey of a stratified patient of TNBC has been, correct me if I'm wrong, Joe. So for the past decades, I would say there's a very limited success in stratifying TNBC patient. We want to treat them differently, but there's very limited success. We don't think we can beat that problem with our study, but we do believe that we're pointing to the right direction, not only with the right biological knowledge, but also the way we're doing that. As Joe mentioned, previously in clinics, people are using this histology images to classify patient without knowing why. And on the other hand, researchers are studying these genes and pathways and cells without knowing, the impact of them in clinics. We're sort of bridging this two extreme side. We're combining, and the power is to bring all modalities together, molecular, cellular, and histoimages all together, and plus the power of AI tools. This is a really, the right way we believe in the future to do study, which will be beneficial not only for TNBC, but for all different studies. This is just, we believe it's the beginning of some new ways of doing research, but we're not getting to that magic time yet.
Thank you. That's a really wonderful answer to that question, and I think it'll set people's mind at rest and also just give us hope for the future as well, which is wonderful. So I'm going to change tack a little bit and ask you about what it's like to work in a multidisciplinary project, because obviously there seem to be many PIs working on this project from many different departments and institutions. So tell me, what challenges and opportunities did working in such a large multidisciplinary team give you?
I'll go first if that's all right. So I have to say I've never worked in a project like this at all. I've never had so many different expertise at hand, and also so many different challenges there. And I do give a huge credit to Heather Ashmore, who was our study coordinator, and she religiously put in monthly meetings so that we could all get together, sit in a room and really talk about where we were as part of the project. So making sure that everyone's on the same page once a month is extremely important to make sure you're all moving in the right direction, and making it clear why certain people need to do certain things at certain times. I think as well, particularly the people in this call, we've been very enthusiastic and very excited by this project, and we're working together. We're very respectful about each other's knowledge and how we can't be experts in every single field all at once. We have to work together to be able to do all of this. So I think the teamwork comes from a shared vision, and also making sure that you're keeping in touch and having that regular communication.
Yeah, did anyone else want to say anything on that one?
As the sort of non-scientist in the team, I was a scientist a very long time ago, but I can't really count myself as one anymore. I think everybody was really good at explaining exactly what their role is, what the techniques are. And as Kui has mentioned, the amount of data that's come out of this is enormous. So the amount of times Akanksha and Alecia-Jane have very patiently explained all the different figures to me, so I can understand what's coming out of it, has been amazing. I think also just the interest in the clinical side of it and wanting to learn about the clinical side of triple negative breast cancer. And we arranged for Alecia-Jane and Akanksha to come and shadow and sit in on some clinics and see how things work in the Precision Breast Cancer Institute as well. So I think that was really great as well. And I think the other side of the project was actually the LEAP program was a massive thing. So there were 3 projects internationally working on triple negative breast cancer, but there was also projects on tuberculosis and glioblastoma. And every 6 months, was it every 6 months or every year, they'd have a whole Wellcome LEAP meeting where we'd all come together. And just learning about those other diseases as well was amazing. And, you know, you had that sense, as although we were a multidisciplinary team, by the time you'd got everybody from the other programs together as well, it was a really amazing opportunity to learn from other disciplines and other teams and other institutes across the world.
Yeah, it sounds like just a wonderful, wonderful opportunity, so inspiring. And you sort of took, you know, inspiration from each other and from the other people in the projects, and you've just created something absolutely brilliant out of it. So it's a wonderful story in that respect. Can I ask, are there any publications forthcoming?
Well, actually, yeah, I'm very happy about this, because just yesterday we submitted the first paper after a very long process.
Great.
Yeah.
And when can we read it, do we think?
Well, I don't know. Hopefully very soon, but it'll depend on the reviewers. Yeah, the paper is focused on the model prediction. So both how the model was built and which therapies the model recommend, and some validation using the data that my colleagues analyzed. But that's only the first part of the story. We are working on like several more publications, which we are very excited about.
Brilliant. I will keep touch with you all so we can keep adding them to the information with the podcast as they come out. So I guess my last, it's kind of a 2-part question. My last question to you all is, what have you all personally gained from being a part of this project? And where do you see your research taking you in the next 5 to 10 years? So I'm just going to go around again. So Akanksha, let's start with you.
So what I gained this lovely team. They are wonderful people to work with. We were very well coordinated. Our discussions were very, very fruitful. We learned new things with every discussion. And I don't think so we ever had a heated argument. I don't think so. So that's how the team was. And for me, it was my first postdoc. So jumping in with such a huge project with that many PIs and such a big team, it was overwhelming in the beginning, but I'm really glad that I started my postdoc journey with this project and this team.
And what do you see yourself doing in the next 5 to 10 years, Akanksha?
Continuing in cancer research and working on the clinical aspect more, because I feel that really helps you understand and bridge between clinic and research. So I would really like to focus on clinical research in the next 5 to 10 years.
I'm just laughing at that because Akanksha's had so many ideas of what to do over the last 3 years. It's quite astounding. So, I think it's generated so many different ideas for different people about where they want to go next. So, yeah.
Which is marvellous.
It's hard to keep up with, but...
Brilliant. And Kui, tell me what you learned, what you personally gained, and where you see yourself in the next 5 to 10 years.
Yeah, so I learned a lot. So the first thing, maybe the less important thing, is how to handle all different modality data at a cohort level. It's the first time, most of the data I processed those data before, but never at this scale, this type of data. And all together, that's the first time I learned all this experience. And I learned a lot of biology as a people who are from the computational world. And most importantly, as Akanksha mentioned, I learned how to work with people. So people from a different background, like from biological and clinical background, they speak different languages, and they think about different things. And also, people that have different ways of working, and how to handle things. Like Alecia-Jane mentioned, how if your PI doesn't cooperate, how do you deal with them? Things like this. I think the most valuable things, as Akanksha mentioned, I think we are not only collaborators but also friends. Besides the parts, non-scientific part, we are quite close with each other as well. I'm quite happy for that. About research, so I think I want to do computational models. So when people say AI, it sounds like a one thing. You build just one thing that solves all the problem like ChatGPT. It doesn't work like that. So for this study, what I learned is if we want to beat cancer, which is very complicated, what we need is, now the way of studying is sort of changing. It's becoming more and more data-driven. So what we need is very deep data from individuals, different type of data from individuals. And more importantly, we need a very wide coverage of the cohort. You need to get that data from different people because people are just different, so that you could identify. I recall there's a title called Unity of Heterogeneity. So you need. There's a title. This is the title of a paper. So which brings to my research. I think first of all, how to deal with this very deep data. So you need models to integrate this data across different modalities. That's the first thing I want to do. More importantly, I want to develop a method that could be cheaply scaled to a large scale, enable people to find those unchanged patterns behind all this data, something that is really influential to clinical studies. So that would be my ultimate dream. So to summarize, I want to eventually bridge this molecular data, scale data, the clinical data, and the biological side and the clinical side. That requires a huge collaborator across the world, across the people from different area. I just want to be part of that.
That's a wonderful aspiration to have. So I applaud you. Pedro, tell me about you. What did you gain from the project and where do you see your research in the next 5 to 10 years?
Yeah, First of all, I agree with Akanksha and Kui that the best thing about this project was the group of people. For me, it was also my first postdoc. And at the beginning, I was a bit scared of being like thrown into this huge project with a lot of acronyms that I didn't understand and a lot of different, moving parts that I had to get into my head. But I couldn't have wished for a better group of people. They are not only amazing, extremely intelligent scientists from who I have learned a lot, but also an amazing group of people. It's true that we have never fought. We have always supported each other and respected each other's expertise. And that has been, for me, the opportunity of a lifetime. Because also I have learned so much from having such a multidisciplinary group of people. Sometimes when I was working in other projects, and you get a data set and you have to analyze it from, I guess my work is a bit more theoretical than clinical. You are not able, you have to guess or fill in the pieces for a lot of gaps in your knowledge. But here, I was able to speak to the clinicians, to the pathologists, to the people who collected the sample, who analyzed the data. So being able to just go to this person and ask, how did you do this, or why do you classify things this way, was extremely valuable and allowed me to have a closeness to the data that I wouldn't have gotten other way. So that has been incredibly valuable. And as for where I see my research going on in the next years, so I would like to keep focusing on doing this kind of cutting edge research to improve breast cancer patient outcome and survival. Specifically, I would like to focus on cancer evolution and treatment resistance, because now we know, of course, that tumors are not homogeneous, they are heterogeneous, and they are subject to evolution. And taking advantage on this to develop new drugs and improving survival times would like to be, and to further personalize our treatments. That's what I want to focus on in the next steps of my career.
Brilliant. And Alecia-Jane?
I've learned a lot of things during this project, I have to say. As everyone has said, it's just been amazing to be part of such a powerful, focused and amazing team of experts in different fields. So I really enjoyed that. And I've never had the privilege of working in such a close-knit team, so that's just been phenomenal. From the PIs, I have to say, what's kind of shocked me is the bigger perspective they have. They say, if you take a team this big, you take this much money, this is what you can achieve. And at the beginning, I really didn't believe that was going to be possible. And I'm just so proud of what we've been able to do over these 3 years. It's far exceeded my expectations. We've all far exceeded my expectations. And so that's just been fantastic. I've learned a lot about breast cancer along the way. So my background's in kind of normal mammary gland biology. And where I see myself taking this in the future is I've just set up the Cambridge Lactation Lab. So I'm actually trying to understand the normal side of the breast. So we've been exploring a lot of the abnormal side. We've been using multi-omic analysis on patients. And I think this is really important to get this multi-level understanding of how certain tissues change in either an aberrant or normal function. So very excited with what we've done, and I'm very excited of where's next.
I mean, I am just so overwhelmed by how brilliant you all are to start with, and it has just been the most wonderful conversation. Your enthusiasm is beyond, and your knowledge and expertise is just phenomenal. So I'm just going to wish you all huge amounts of luck. I hope you still get to work together on future projects, because that teamwork element is just so wonderful to hear and so fantastic. And I'm going to give the last word to Jo.
I think it's been amazing. I was really lucky to be involved in some of the discussions for the grant application for this, which was all done, as grant applications all seem to be, in an amazingly short amount of time, and with maybe not a massive amount of expectation that we'd actually get the funding. So yeah, it all happened really fast, and it was really early on in me being a program manager as well. So I think it was actually ideal, because it made me move from my comfort zone on the clinical side and clinical trials into the more laboratory-based research. I got to know, some really amazing group leaders and researchers as well. And I think for someone starting off as a program manager, wanting to sort of understand the world of multidisciplinary research in a place like Cambridge, actually getting involved in a project like this was a godsend, really. It was just the perfect way for me to really get into my role and see who the key players were around Cambridge and London. It's been truly amazing.
Well, thank you all so much for joining me this afternoon. That has been just a lovely way to spend an hour hearing about your wonderful project and your brilliant research. So I wish you all good luck and thank you very much.
Thanks so much. Thanks for having us.
Thank you for having us.